about time to start.
Okay.
Okay, thank you. Please.
So wait 1 minute. It's setting up.
Okay, good.
Okay, now we are on YouTube now.
Okay.
Okay. Should I start now?
Yeah, if you are ready, you can start.
Okay. Hello, everybody. Here is Xin speaking. Today, we are honored to have speaker Professor Xizhuo Jiang from Northwestern University of China. Xizhuo is a full professor at mechanical engineering in Northeastern University, China. She obtained her PhD 4 years ago, 2018, from University College London, UK. Her supervisor was Luo Kai, is a fellow of Royal Society of Engineering, and also Professor Yanis Vandikos is also a fellow of Royal Society of Engineering. Both of them were supervising Xizhuo. She then worked as a research fellow in University College London for more than 2 years. Before her PhD, she obtained her Bachelor of Engineering and Master of Engineering degrees from Tsinghua University, one of the best universities in China. She had 2 years engagement in industry as a mechanical engineer from 2010 to 2012, and worked as a research assistant in Tsinghua University from 2014 to 2015. So Xizhuo was both engineering and a scientist, so had a lot of experience from both industry and academy. So today, we are very honored to listen to his talk on the title of Molecular Dynamics towards understanding atomic events on endothelial cell surfaces. Okay, please, Professor Jiang, go ahead.
Thank you very much. I'm going to share my screen. Can you see my slides?
Yes.
Okay, very good. Thank you. Thank you, Xin, for your introduction, and thank you both Xin and Li Zhen for inviting me. Good evening, good morning, good afternoon, everyone. I'm Xizhuo from Northeastern University, China. So I think when you hear the name of my university, some of you may find it's quite confusing. I guess when you listen to this name, what comes to your mind is the university in Boston. As far as I know, at least 3 universities, the name of which has the meaning of Northeastern, and ours is one of it. Unmute. Can you hear me? Sorry, can you hear me? I found I was muted just now.
Yes, now it's good.
Okay, good. So my university is located in the northeastern part. It's located in Shenyang, metropolis in the northeastern part of China. So I hope you won't be confused from now on. Okay, let's come to our topic today. So the title of my talk is Molecular Dynamics Towards Understanding Atomic Events on Endothelial Cell Surface. So when you read the title, you may wonder, what is endothelial cell surface? So now let's start from endothelial cell surface. Endothelial cell surface, sometimes we call it endothelial surface. So the upper left figure is a cross-sectional part of our blood vessel wall, and this is the lumen side. Our blood vessel wall consists of a single layer of endothelial cells, and on the surface of endothelial cells is covered by a layer of hairy structures like this, and this hairy structures is called endothelial glycocalyx. Glycocalyx is in direct contact with blood, is in the lumen side, and the malfunction of glycocalyx usually causes some diseases like cardiovascular disease or renal disease. The hallmark function of glycocalyx includes mechanotransduction. Mechanotransduction means the glycocalyx senses mechanical forces from the blood flow and then transmits the mechanical signal into the cytoplasm, and then our cells react to the flow, and we can have some biochemical processes happen. And another function of glycocalyx is it works as a microvascular barrier. So it means the glycocalyx controls the permeability of the blood components. In previous theoretical study of mechanotransduction, the glycocalyx as a whole is regarded as a rigid body. So when flow passes by, so the flow shear, there will be a local torque, and the flow shear stress first transmits to the tip of the glycocalyx, and then to the fork-like structure of the glycocalyx, and then to the cytoplasm. And there will be a deformation of membrane, and then the signal is activated. In a previous calculation, it is found that the wall shear stress near the endothelial cell is enhanced in the presence of glycocalyx, and is about 1 to 100 times larger than the flow shear stress. When people investigate the microvascular barrier role of the glycocalyx, a reversed Starling principle is always used to quantify the interstitial fluid exchange. And here I put the formulation here. So the problem of such a formulation is that it only considers the static state of the glycocalyx. In other words, that means the effect of blood flow is not included in this term. When we talk about glycocalyx after my introduction, I think when you listen to glycocalyx, you may think, oh, it's a type of biological thing. Yes, it is. So we know that in biology, people prefer to use experiments to conduct this research. However, for glycocalyx, experiment is extremely difficult. Of course, the glycocalyx on the endothelial cells, they are highly dynamic and fragile. So when the endothelial cells are taken out of our body, when they come to our laboratory, so the glycocalyx will become unstable. The instability sometimes cause the biased conclusion. For this reason, some scientists turn to numerical methods to get some inspiration. So here we searched in Web of Science with specific index. And after peer review of relevance, we found that there are 43 papers talking about how numerical methods can be used to solve endothelial glycocalyx related problems. And if we look at these papers, we found the trend is, how people use the glycocalyx, the trend is very similar to the history of the development of numerical methods. The first studies about numerical methods to solve EG-related problems they were using mathematical models or CFD, FEM, some continuum-based. And in 2010, lattice-Boltzmann method, dissipative particle dynamics have been used to investigate EG-related problem. And before that, some simple Monte Carlo method, some simple molecular dynamics models were built. And at 2015, actually something happened, and we can find that all at large-scale molecular dynamics have been applied to the endothelial glycocalyx to this area. Here I want to mention, I'm very happy to mention a paper. It talks about the concurrent coupling of dissipative particle dynamics and the molecular dynamics. And the host of our CFSM seminar, Li Zhen, is one of the co-authors. To be honest, this paper is one of my favorite paper. I can remember almost every detail of this paper. I read it quite a lot of times. And here I show the number distribution of the numerical methods. And we found that in terms of number of papers, molecular dynamics is now a very powerful tool to investigate the interesting phenomenon in the endothelial glycocalyx. Okay. So some of you, if you don't do molecular dynamics, you may not know what it is. So here I use one slide to briefly introduce this method to you. The governing equation of molecular dynamics is Newton's law of motion, F equals ma. And if we know the force on every individual item, theoretically, we can work out the acceleration. And after integral, we can get the velocity. And integral again, we can get the displacement of the position with some initial conditions. So now we have to solve one problem. How can we know the force on every individual item? Oh, it's really a big problem. So how people do that? We know that force is also the negative, the positive of the gradient of energy of potential. So now we switch the problem to how to work out the potential. We do have a few ways. So a popular way is you will use some parameters to estimate to quantify the potential. And the set of parameters that we use to calculate, to quantify the potential is what we call the force field. And there are some popular force field I just name a few, like CHARM, OPLS, AMBER. As molecular dynamics is so popular, so some great developers, they have offered some great platform that we can use directly for our MD simulation. For example, NumD, LAMS, and GROMAX. So this interesting animation in the corner is actually what we have simulated use molecular dynamics, and this was done by my master student. She is in the seminar now. I know. Our aim is to use the molecular dynamics to study the EG functionality, and we hope to see some new insights by doing molecular dynamics. And to be specific, we know that the glycocalyx is hairy structure, and we have a sense that it is quite flexible. So the first thing we want to work out is what is the flow profile through the tree-like structure over the endothelial glycocalyx. And after that, we want to know the mechanism for mechanotransduction. And last, we want to give a more accurate estimate of ion transport through the endothelial glycocalyx layer. So as I mentioned, we want to use molecular dynamics. As the name implies, we need to know the molecular structure of the glycocalyx. So here I show a unit of glycocalyx. This is the lipid membrane. It's part of the cell membrane, part of the endothelial cell membrane, and the red beads represent the coprotein. Coprotein is actually a protein that these hairy structures can be attached, can be anchored to the cell membrane. And this green change is called glucosaminoglycan change. That's quite a long name. So usually we call it a GAG change. If you can't remember, just remember that that is sugar change. Yeah. Okay. If we look into the if we look at the transmembrane part of the core protein, so it's actually a list, a sequence of amino acids, and I put the amino acid sequence here. And if we look at the chemical composition of the glycocalyx chain, the sugar chains, so here one type of the GAG chains that is ubiquitously found in our body is HS chain, hypersulfate chain, and this is chemical potential. You don't have to remember that, just keep in mind that HS chain is of high negative charge. Yeah, and this is also the feature of glycocalyx. With the molecular model, now I can build my MD model. So this is the glycocalyx and a patch of the cell membrane. And all the biomolecules are solvented in a solution of sodium chlorate with a concentration of 0.1 mole per liter. And this one is the lipid membrane, as you have already seen. And above that, for this region, we have the main structure of glycocalyx. And in this part, we use it to simulate the lumen part of the blood flow. That means the lumen part of our blood vessel. That means the flow will pass it by here. And below the lipid membrane, it is just the cytoplasm part. We focus on the lumen part. And in terms of the size, the height of the MD domain is about 72 nanometers. To be honest, it's quite big for biomolecule simulations. And in this research, we want to investigate the behavior of glycocalyx in different flow conditions. So the flow will only impose X direction flow, and to mimic flow, we impose external force on water oxygen. By varying the magnitude, by varying the strength of the external force, the velocity can be changed. So for clarity to this part, for this model, I only show the biomolecule. I only show the biomolecules. So the platform I use is NumD, and VMD is used for you for the VMD is used for the visualization. Well, for my system, as I mentioned, it's quite big. It includes about 6 million items. And for a typical case, the physical time I simulated is about 30 nanoseconds. And that's quite a monster. And to harness such a monster, I have to employ 9,000 cores, and 9,000 cores have to work 8 hours to get a physical time of 5 nanoseconds. This work was done when I was in the UK, and the high performance computer I use is Archer. But Archer is now out of date. Archer 2 has already taken over the role of Archer. Archer retires. Before I show you the result, I'd like to share the computational cost that such a big MD simulation involves concerns. For such kind of research, we have spent almost 300 million core hours. That is equivalent to 10,000 cores, continuous working for more than 2 years. Yeah, you see the computers working, yeah. And I have to mention that the data we have to propose is quite colossal, and it's about 40 terabits. If you like classical music, if you like Beethoven, you know how long it takes you to finish Beethoven's 9th symphonies. For example, this one, Karajan's, 40 terabytes equivalent to use your mobile data to play Beethoven's 9 symphonies 20,000 times. Wow, that's quite huge. A lot of money, a lot of mobile cost. Okay, it's just a joke. Okay, let's come back to our results. If you remember, the first objective of our research is to investigate how the flow is like in the presence of glycocalyx. And we have analyzed the flow profile over the height of the glycocalyx, and we see that the velocity profile are always oscillating along the height in the presence of glycocalyx. And we also calculated the shear stress, and we found a hierarchical distribution of shear stress, and the shear stress near the cell, near the endothelial cell surface is enhanced, which agrees with previous study. We also looked into the dynamics of the glycocalyx. If you remember, we only impose X direction flow. However, the heterogeneous conformation of the glycocalyx convert the X direction flow into the other two directions. So from the figure, we can see the glycocalyx can swing in the X direction. This is not surprising, yeah. And we can also find the swirling motion of the glycocalyx of the sugar chains. So here I want to mention an interesting phenomenon. Please pay attention to this one. For this sugar chain, at the very beginning, it has a coiled structure. However, when the flow passes by, you see what happens. Okay. Can you see that? The
Yes.
coiled structure uncoil while wave motion. Oh, that I will talk about the biological significance later. Okay, before we move on, I think we have something to discuss. That is the magic of MD, and that is new insights brought by MD. So at the very beginning, I mentioned that in previous study, they mentioned that the false transmission is transmitted from the tip of the glycocalyx and then to the fork-like structure. And now we know that is the sugar chain. And then the false will goes into the coprotein and finally arrived into the cytoplasm. This is the traditional way of false transmission. However, our molecular dynamics simulations give you some different alternative results. This is the motion, for example, the purple one, the pink one is about the movement of the protein, and the other parts, the other lines are about the movement of the sugar chains. We found that the motion of sugar chains and the proteins, they are not synchronized. So they just move in their own ways. They don't relate. They are not related. So that implies that an alternative false transmission way is here. So the false can be transmitted directly to the coprotein and then to the cytoplasm without visiting the sugar chains. And this is the alternative pathway that is revealed by our MD simulation. Okay. From previous animation or slides, you have impression that the glycocalyx is not rigid. It's not. It's something like a soft matter. So the first thing we do is to demonstrate it is soft matter rigorously. So I won't go into the detail. I just put the conclusion here. So we have demonstrated that the glycocalyx behaves in a soft matter way, not a rigid body. Okay. So the next question we want to ask, if glycocalyx is not a rigid body, how could the soft matter stuff transmit force? So now we look into the dynamics. We focus on the dynamics of the transmembrane part of the membrane. The transmembrane part of the core protein, and we found that when flow passes by, the X type, the cedar-like structure, the cedar has been closed by 6 degrees. So we have impression that the glycocalyx or the core protein may transform force in a cedar motion, but this is just our first speculation. So next, we use principal component analysis to demonstrate it. After the principal component analysis, we found that the motion of the transmembrane protein, the principal motion is the cedar-like motion. It occupies more than 50%. And the second and third important motion is bending in 2 directions. So we can conclude that force is mainly transmitted via a cedar-like motion, but plus some bendings. We know how the force is transmitted. And the next question we want to ask, how strong? What is the strength of the force? To work out this problem, we used another molecular dynamics technique. It is called steered molecular dynamics. What does it mean? Actually, it is very similar to atomic force microscopy. What we have to do is we impose force on one end of the core protein, of the transmembrane part of the core protein. And we pull this end, and to keep this end moving at a constant velocity, but the velocity is very, very slow. And we know the cedar at the very beginning has an angle of 56 degrees. And what we do is we slowly close the cedar until it reaches 50 degrees. And then we record the force that we use to maintain the constant velocity. And then we draw the force distribution. And the force distribution showed that the order of magnitude of the imposed force is 10 to 100 piconewton. And that means the force transmitted into the cytoplasm also have the same order. And we have also compared this magnitude, this order of magnitude with other similar biological phenomena, and we found that they agree well with each other. So next, we have studied what is the role of the sugar chains. And the method we do is we just pull out some sugar chain at different locations and see what happens. And the conclusion is that for the core protein, there are some sugar chains, growing on the core protein, and also there are surrounding sugar chains. And we found that the central sugar chain that grows on the core protein will enhance the deformation of the core protein, but the surrounding sugar chains will inhibit the movement of the central core protein. That means the surrounding sugar chains will form a protection for the central core protein. Yeah. So now here I use one figure to summarize the new findings that is revealed in mechanotransduction by molecular dynamics. Okay. So our last objective is to investigate the ion transport and if possible, we may revise the Starling principle. So if we want, so here we use sodium ions as an example. If we want to investigate how ion is transported across the endothelial glycocalyx layer, the first question we have to answer is, what is the direction of sodium ion transport? To answer this question, what we have to do is to work out the sodium distribution along the glycocalyx, along the head of glycocalyx layer, and we found that this layer is beneath the dense part of the glycocalyx. This layer is near the cell surface, and this layer is in the flow region, and we found that the layer beneath the glycocalyx near the cell membrane, the sodium concentration in this layer is always higher than that in the main flow region. So we can conclude that sodium ion is transmitted from the inside of the glycocalyx to the main flow region. We know that after that, we have changed the velocity by varying the external force of water oxygen. And we found that when we include the blood flow velocity, there are more sodium ions. More sodium ions will be carried out of the glycocalyx region to the blood flow region. Okay. So we want to ask how it happens. How come? So we do another experiment. So in this experiment, we use two sugar chains. They have identical chemical compositions. The only difference is the conformation. For this one, A01B, it has a stretching structure, and for B01B, the sugar chain is coiled. This is the only difference. Otherwise, they are identical. So what we have to do, we calculated the number of sodium ions around these two sugar chains, and we found when the simulation goes, when they change the structure, the number of ions surrounding the stretching sugar chains is fewer than those around the coiled structure. That means the coiled structure tries to confine the sodium ions in its own realm. If you remember at when we study the animation I show you, when flow passes by, the coiled structure tries to uncoil with the waving motion. So when flow passes by, the glycocalyx, the sugar chains, they prefer to stay in a stretching state. So that explains why the previous conclusion that when the blood flow velocity increase, there are more sodium ions being carried out of the endothelial glycocalyx region, and they are carried out to the blood flow region. Okay. So just now we know how the flow affected the ion transport. So we will come back to the Starling principle. So here we introduce coefficient gamma to represent the effect of blood flow. And by using our simulation results, we give the expression of gamma. And when including gamma, we found that in a normal state, the accuracy of the osmotic part of the filtration force can be, the accuracy can be increased by 8%. And this helps us to understand more about interstitial fluid exchange. Now let's come to a very interesting discussion. I will have a question here. So this is a movie recorded by a previous biologist. And this is our blood vessel walls, and this moving stuff is the red blood cells. We can always see there is a gap between the moving red blood cells and our blood vessel walls. And the previous study showed that when we increase the blood flow, the gap is increased. The gap increases. There are a few papers talking about this phenomenon using fluid mechanics methods, which I totally agree. But today I want to explain this phenomenon from a different angle of view. If you use the red blood cells, the surface of red blood cells is also covered by glycocalyx and is of negative charge. And we also know that the blood vessel walls is covered by glycocalyx. So both have negative charge. So they have repulsive interactions actually. And we know that when we increase the blood velocity, more sodium ions will be thrown away from the endothelial glycocalyx. So that means the repulsive force between the red blood cells and the endothelial glycocalyx increase. So that explains the increase in the gap between both. This is this is an explanation for this phenomenon from a different point of view. Actually, a faster blood velocity will benefit our health because red blood cells carry nutrients, carry oxygen, and a slightly faster blood velocity will promote our metabolism. So now I want to ask a question. How could we increase our blood velocity? Okay, you just keep your answer in your mind. Our suggestion is that we suggest you to exercise regularly. Apart from that, we also encourage you to reduce salt intake. The reason is, you may wonder, I think you think, oh, it doesn't matter whether we have too much salt intake or not, because our kidney will help us digest it. However, the literature also shows that if we take in too much salt, only part of the salt can be digested by our kidney. The remaining will stay in our blood vessel and will stay in endothelial glycocalyx layer. It means the repulsive interactions between red blood cells and the blood vessel walls will decrease as we have some positive charge from sodium ions. Phenomenological understanding is that the red blood cells becomes sticky. So in terms of that, we also suggest you to reduce salt intake, and don't forget to exercise regularly as well. Yeah. So just now I have mentioned some new insights revealed by molecular dynamics. However, when we were using molecular dynamics to do glycocalyx related topics, we always face different types of challenges. Here I just list a few. So the first thing we have to face, the first challenge we have to face is how to construct the glycocalyx, as you know, as you have impression that it's quite fragile, it's dynamic, and also difficult to find an accurate model of glycocalyx. So by far, we only have one type of glycocalyx. That is not enough at all. We still need more models to come. The second challenge is about the force field. As far as I know, force field about protein and the lipid membrane, they are developing very fast, and we have sufficient data to validate. However, when it comes to sugar, to carbohydrate, the thing is not easy. We're still lacking force field about sugar, about carbohydrate. The third problem is the in silicon capability. To cover the sheer size of glycocalyx, we really need lots of computational cost. It's costly, and if we want to do simulations of, if we want to do molecular dynamic simulations, we need sufficient funding to support us. And the last thing, large scale simulations always means a colossal set of data, and how can we accurately analyze the data is also a challenge for the young scientists, especially for beginners. Okay. So this is almost the end of my talk today. So here I want to use use this figure to summarize all the new findings that molecular dynamics have given to us. I really enjoy working in molecular dynamics, and also I want to briefly share my ideas about how MD will develop in the future. As I mentioned, MD simulation, especially fully detailed MD or all-atom MD, is quite expensive. So a way to reduce computational cost is we use some coarse-grained method. You know, not always we need all the information. Usually we only need part of it. So maybe we can develop some coarse-grained method. And we also know that in the world, nothing in the world is a single scale. Most of the problems are multi-scale. So we need MD to couple with other numerical methods, especially from larger scales. And the third one, I think MD could be interact with machine learning, and machine learning will give us will open a new door for MD. Actually, in 2020, a group has combined the machine learning and MD. And they have simulated a system with 100 million items with ab initio accuracy. And their research has won the Golden Bell Prize in 2020. Okay, this is my talk. This is all of my talk today. Thank you very much. Thank you for joining today. And if you have any questions, I'm very happy to answer. So this QR code can direct to my home personal page. Thank you very much. Thank you.
Okay, thanks, Xizhuo, very nice talk, very interesting. I think we all learned a lot about glycocalyx. Also myself, I think we partially touched in the past of our research about the glycocalyx, but I never looked into so much detail. So in the audience, if you have a question, please just unmute yourself and speak. Thank you.
Hi, I have a question. Hi, thanks for this presentation. I appreciate it as well. It was very clear and soft to follow. I have a question regarding the glycocalyx. Actually, are they only present at these core proteins for endothelial cells, or they are present elsewhere? For example, in other organs?
It covers all the blood vessel walls. Whenever the blood vessel walls can reach, we can find glycocalyx, and we can also find it in some cancer cells, tumor cells, and red blood cell.
On the
It's
surface of the cell?
Yes, the surface of tumor cells. It's almost everywhere.
Okay, and why the other cells do not fix the glycocalyx? You have an idea? The normal cell?
Because endothelial cells occupies almost most part of our cells. So we focus on the glycocalyx of endothelial cells. And for cancer, for tumor cells, the composition of glycocalyx is quite different. And we haven't got any information about the chemical composition of that part. So that's why I didn't go deep into that one. Yeah.
Yeah, thanks. And then I have another question regarding the sodium ion simulation in MD. Do they have specific beats that you follow, or you take them into account with a force field, with a specific force field?
Okay, thank you very much for your question. This is really good. For sodium ions, I think the force field is quite mature. The parameters are well established. And usually different force field can share the data for sodium ions. No matter whether you use in terms or the other ones, the data are very close. Yeah.
Okay, thanks a lot again.
Thank you.
Any questions? Any more? Shizhuo, I have one very quick small question. So let's say I'm a CFD guy. I come from continuum. So I want to really study flow inside the blood vessels, veins or artery, whatever. But I don't have the computer power to really simulate this full detailed MD. So do you have some suggestion? How should I keep some component to cross-grain the essential part? So I really would have a dynamic simulation of CFD and then properly capturing the boundary condition of glycocalyx effects.
Well, I think you can regard glycocalyx as chain of beads. You don't have to go into details. You don't have to separate as sugar chains or copper. You just regard it as a polymer. Yeah, just as I think previous study usually treat glycocalyx like that, and I found it works well.
Okay, that's cool.
Okay, thank you.
So Shizhuo, can I ask a question about the you mentioned we need reduce our sodium salt intake. I'm wondering for healthy part, should we have the distance between the blood cell and the endothelial cell it's smaller, or we want to increase the distance when they flow? So you mentioned if we reduce the salt intake, we can reduce the sodium concentration, so that the electrostatic force will be weaker, right? I'm not sure. We want to increase the distance, or we want to.
I see.
Reduce
yeah.
it.
I understand. I know what we are talking about. I read a paper from a German biology or physiologist, and he suggests us we actually need a slightly bigger distance between the red blood cells and the walls. Otherwise, it seems that our red blood cells become sticky. So if it moves slowly, it's not good for our metabolism. Yeah. So just ask it to move faster and reduce salt intake. Yeah, for that reason. Yeah.
Okay. So the suggestion is take more exercise.
Yeah, exactly. Yeah, if you taking too much salt, just jogging and sweating, yeah, and I think it should be fun. Yeah, okay. I think the conclusion is quite common, but it's the first time we look at this question from the molecular level. Otherwise, it's all about the epidemiology aspect. Yeah.
Yeah, it's very important to explain the concentration and sodium ion concentration change over the flow, different flow condition. This is very important to help us understand why we want to do this.
Exactly.
Yeah.
Okay.
Thank you.
Thank you.
Oh, hi, Xizhuo. This is
Hi.
Zhao Yan from University at Buffalo. So I'm very happy to listen your talk. In fact, it's more like some voice from my hometown. So my hometown is Xinjiang. So
Wow.
yeah.
Good.
So and a very interesting talk. So I do have a question about your force field. So you present a very kind of high resolution, like atomistic scale of your system, like the long chain, like molecules, and also the bilipid bilipid like membrane. So I'm wondering, so when you try to validate your force field to describe those kind of membrane or like polymers, what kind of like properties you think which is more important? So obviously you cannot like capture all like behaviors and the properties of the polymers and the membranes. So, but I'm wondering what is the important like properties you want to make sure that your force field can capture?
Okay, thank you very much for your question. Actually, it's a very popular question that people would like to ask me. First, I use some well-established force field. For example, for the copper, for the protein, for the lipid membrane. They are well established and they have been validated from all aspects, from experiment or either from quantum mechanics calculation. I think people focus on their biophysical properties, not biochemical properties, like how the deformation comes and how the conformation change, something like this. And a special thing for my research is about the sugar chain, because it is sugar, it is carbohydrates, and it's not, I mean, the parameters for force field is limited. So, so when we selected the force field, we also focus on the biomechanical aspect of the sugar chain. For example, the binding rigidity, stiffness, binding stiffness. And another thing we focus on is the solubility. I mean, how well it can be solved in water. Yeah, this is another aspect we focus on. I admit that when you select the force field parameter, it really depends on what aspect you are looking for. There are quite a few options. So yeah, we have a procedure of selection. Yeah.
Yeah, thank you very much.
Thank you.
Xuejing, you have a question?
Yeah, hi, Xizhuo, nice talk. I have a question, a quick question. So I'm wondering what's the mechanical property, like the elastic modulus of the glycocalyx layer and its component sugar chain. So
okay,
how do you model them in your simulations? Because you use the MD simulation. It's quite detailed simulations. Yeah.
Okay, thank you very much. For the biomechanical properties, for example, the binding stiffness, actually we have linked the persistent length to the binding stiffness. There is a statistical formulation for that. And what we have to know is about the end-to-end distance for MD. So according to that statistical formulation, we only to work out the end-to-end length. And
so
in our
like chain model.
Sorry?
I mean, in your MD simulation, you use a warm like chain model or not? WLC. No?
Oh, yeah.
Yeah. Oh, go ahead.
Yeah. And in our simulation, to get the end-to-end length, we have to do quite a few replicates, and we draw the distribution of end-to-end length, and we get an average, ensemble average, and then put it in the WLC model, and then we get the persistent length, and then we calculate the binding stiffness.
Okay,
Yeah.
thank you.
Is it clear?
Yeah, clear.
Okay, thank you.
Qing, are you still here?
I'm here. I don't know. Should I say a closer word? Okay, it's getting late. If there's no other question, so let's thank Xizhuo again. I think you will not hear the applause, so we'll just share it up in our head. Okay. And hopefully we'll meet soon in person. Okay. Thanks again. Thank you. Have a good night.
Thank you. Goodbye.
And have a good morning
Xizhuo.
for the people in the US. Okay.
Bye bye.
See you.
Bye.